Today, we are discussing a fascinating study titled Hepatic malonyl-CoA synthesis restrains gluconeogenesis by suppressing fat oxidation, pyruvate carboxylation, and amino acid availability. This study uncovers an unexpected role for malonyl-CoA in regulating glucose production in the liver. To understand the importance of this study, we first need to review what malonyl-CoA does. Malonyl-CoA is produced by the enzyme acetyl-CoA carboxylase, or ACC. In the liver, malonyl-CoA serves two main functions. First, it acts as a substrate for de novo lipogenesis, the synthesis of fatty acids. Second, it inhibits CPT1, the mitochondrial transporter that allows long-chain fatty acids to enter mitochondria for oxidation. Because of this dual role, malonyl-CoA functions as a metabolic switch. When malonyl-CoA levels are high, fatty acid oxidation is suppressed and lipid synthesis is promoted. When malonyl-CoA levels fall, fatty acids enter mitochondria and are oxidized for energy. Traditionally, malonyl-CoA has been viewed mainly as a regulator of lipid metabolism. However, the authors of this study asked whether malonyl-CoA might also regulate glucose production in the liver. To answer this question, the researchers used two approaches. First, they created mice lacking both ACC1 and ACC2 specifically in the liver. Second, they used pharmacological inhibitors of ACC. These models allowed them to reduce hepatic malonyl-CoA levels and observe the resulting metabolic effects. As expected, reducing ACC activity lowered malonyl-CoA levels and activated fatty acid oxidation. This was evident from increased ketone production and elevated markers of mitochondrial fatty acid oxidation. But the most surprising finding was that reducing malonyl-CoA actually increased gluconeogenesis. The liver began producing more glucose even in the fed state. To understand why this happened, the authors investigated metabolic fluxes using stable isotope traces and metabolic flux analysis. These experiments revealed that loss of ACC activity increased TCA cycle activity and pyruvate carboxylase flux. Pyruvate carboxylase converts pyruvate into oxaloacetate, a key intermediate required for gluconeogenesis. Importantly, pyruvate carboxylase is activated by acetyl-CoA. When fatty acid oxidation increases, mitochondrial acetyl-CoA levels rise, which in turn activates pyruvate carboxylase. Thus, when malonyl-CoA levels fall, fatty acid oxidation increases. This produces more acetyl-CoA, which stimulates pyruvate carboxylase and drives gluconeogenesis. In addition to these metabolic changes, the researchers found that fasting metabolism was altered in mice lacking hepatic ACC. Normally, glucose levels decline during prolonged fasting. However, these mice maintained higher blood glucose levels. Further investigation revealed that this effect was driven by increased amino acid availability in the liver. During fasting, proteins were broken down, providing amino acids that could be converted into gluconeogenic substrates such as pyruvate and TCA cycle intermediates. When proteolysis was inhibited, the elevated glucose production was normalized. This confirmed that protein breakdown contributed to the increased gluconeogenesis. The study also uncovered activation of the transcription factor NRF2. NRF2 regulates antioxidant and stress response pathways and was strongly induced when ACC activity was inhibited. The authors suggest that increased oxidative metabolism may activate NRF2, contributing to metabolic adaptation. Taken together, these findings reveal that malonyl-CoA is not only a regulator of lipid metabolism, but also an important suppressor of hepatic gluconeogenesis. By inhibiting fatty acid oxidation, malonyl-CoA prevents excessive activation of pyruvate carboxylase, limits TCA cycle flux, and reduces substrate availability for glucose production. These discoveries have important implications for metabolic disease. ACC inhibitors are currently being developed as therapies for fatty liver disease. While these drugs may reduce hepatic lipid accumulation, this study suggests they could also increase gluconeogenesis and alter amino acid metabolism. In summary, malonyl-CoA acts as a metabolic brake in the liver, coordinating lipid metabolism, mitochondrial activity, and glucose production. Removing this brake leads to increased fatty acid oxidation, enhanced TCA cycle flux, and elevated gluconeogenesis. This work expands our understanding of how central metabolic pathways are interconnected and highlights the complex consequences of targeting key metabolic enzymes. Thank you for listening.